Welcome to Living Vividly, the podcast where health, wellness, and curiosity collide. I'm Jackie, the founder of Vivid Health and Wellness, your guide on this journey to living your happiest and healthiest life.
My co-host Daphne, the unconventional dietitian, is always ready to shake things up and bring a fresh perspective to your wellness journey. And we can't forget Dustin, our producer, who keeps everything running smoothly behind the scenes. Together, we'll chat with health and wellness experts along with everyday people who have inspiring stories and practical insights to share. If you're ready to nourish your body, move better, and discover simple ways to thrive, you're in the right place. And hey, if you enjoyed today's conversation, don't forget to leave us a comment sharing your thoughts, and subscribe so you never miss an episode.
Welcome back to another episode of Living Vividly. Today on the podcast, we're joined by Dr. Jacob Breaux, a board-certified gastroenterologist with the Gastro Clinic right here in Lafayette. He's not only incredibly knowledgeable when it comes to digestive health, but he's also deeply passionate about helping his patients understand how their gut health connects to their overall well-being. You may remember Dr. Breaux from our January Health Revolution event on gut health, where he blew us away with his insights and ability to break down complex topics in a way that truly resonated with the audience. From colonoscopies to microbiome testing, he blends evidence-based medicine with a patient-centered approach. And today, we're diving even deeper into the world of gut health, the microbiome, immune function, diet trends, and how it all ties into your energy, mood, and longevity. Thanks for joining us today.
Thank you.
So, Dr. Breaux, the hot topic right now is the gut microbiome. And just we have heard so much about that over the past handful of years, I guess, although many people have been studying it for decades. Can you give us a brief overview of what that means, gut microbiome?
Yeah, and so the gut microbiome, and it's important to think of it as, you almost want to think of it as a rainforest. So it's not just bacteria, it's viruses, it's some parasites, which are sometimes pathogenic, but it's tons of different organisms, including fungi and bacteria and the viruses like we talked about. The biggest thing that you want to have is it's very important for a good microbiome to have lots of species diversity. You want to think of it as, just like I said, a rainforest where you want homeostasis. You want lots of different species to help balance each other out, because that way you can also protect if you have any outside organisms come in. So it's important to have that defense system. And then furthermore, the microbiome communicates with the brain-gut axis. And then you also have lots of hormones and different chemicals that are produced from the gut bacteria. It's a very dynamic system. It has a lot of protective functions, communicative functions, and it's a very cool system.
It's my understanding that we have trillions, 10 trillion is the number that I've heard, trillions of different, not variety, not diversity, 10 trillion diversity, but 10 trillion microbes that live in our digestive tract. And so I always think of it as kind of like a community, that you have to have such a big variety of different personalities like you would in a community, but the microbes work in conjunction with one another, but they're also feeding information to your organs, not just your brain, but your pancreas and your liver and your gallbladder. And there's a lot of cohesive interconnection that happens whenever it's working the way that it's supposed to. I don't know if I don't know that would you say we have the perfect microbiome that we're all trying to get to, or everybody is a little bit different in the way their microbiome is going to work for them?
Yeah, it's very different. It's very person specific. They've had a lot of studies where they try to take and see, okay, we'll take a healthy individual and see what is their microbiome, and they'll do the genetic sequencing for it. The problem with that is sometimes, that healthy person might be healthy in spite of their microbiome. Sometimes the microbiome is assisting in it. The tough thing with people, when you have varying diets, you have varying activity levels, varying hormones, various ages, there's a lot of factors that play into what would constitute a healthy microbiome for that individual. We do know that you want to have more species diversity. For example, some of the microbiomes and hunter-gatherer tribes, especially in tribes in Tanzania and things like that they've studied, they'll have 900, 1,000 species. Whereas the modern American diet, which is high in sugars and fats and things like that, and very low in fiber in relation to what we need, maybe only have 300 or 400 species. So a lot of times we try to focus more on diversity as opposed to, this set of species, because some people will just never have certain species. And even if you try to colonize them with certain types of probiotics, we do know, after 60, 90 days of stopping it, they'll revert back to some of their baseline microbiome. So a lot of it is just more focusing on the health of it, trying to expand and increase that species diversity, but trying to manipulate it to have a certain microbiome, that's more difficult. So a lot of the research has been pushed more towards, making sure you have an adequate amount of fiber, which, you'd love 35, 40 grams of fiber for everybody, but that's tough to work up to. You want to incorporate lots of fermented foods, at least, a serving or two a day of something like kombucha with low sugar. Sauerkraut. Yeah. And so really just trying to focus on, you want a good, healthy, diverse population that has a lot of different species that can kind of serve the multitude of functions that they do. What you don't want is an environment where those microorganisms are not being fed. You start losing species. And we've actually seen, over the last 40, 50 years, there's a lot of drop off of species and a lot less diversity in the population. It happens with aging anyway, but we're seeing where some species are potentially getting lost forever. They're just falling out of the population, especially with the modern American diet. So to kind of circle back to your question, there's not a specific microbiome pattern that we're looking for. It's more lots of species, lots of diversity, lots of health and balance in the system. Like you don't want to, eat fiber for 2 weeks and then go back to fast food for 2 weeks and then eat fiber for a week.
Right.
You want to just, think about it as maintaining the homeostasis. It's like that rainforest. You need rain every day, but not too much rain. And you need just enough. You need just the right temperature and the humidity. So for the microbiome, you need just the right diet, the right foods, the right movement, the right sleep patterns. You're just trying to kind of protect this system within your system.
Okay, question real quick. You're saying it's bad to go back and forth, right? 2 weeks.
It's just not as productive.
Right. But is that better than only not eating the right amount of fiber or whatever?
Anything is helpful. It's just you don't know how long those species will last before you'll lose some, or they'll drop down in a number in the GI tract because there's such rapid turnover. These organisms are dividing, multiplying so rapidly. So say you for 2 weeks, 3 weeks, you don't eat any fiber, like you're going to take a big hit to some of those organisms that might have been depending on whatever it is you're eating. For example, lectin in apples, like there might be an organism that's their food source. So if you just don't eat an apple or don't eat anything with lectin for weeks, that population is going to dwindle down. And then potentially a different population that you don't want might kick up.
Gotcha. Okay, cool. I was wondering, I didn't know if it confused it or if it was just like they just don't survive anymore.
Yeah, just think about it. It's like a zoo. Like you're like, I'm not going to feed the tigers this week and I'm going to feed all the giraffes, and it's like they might survive, but it's just not a good way to keep a balance.
Gotcha.
I think what I've understood is that we basically kind of get our microbiome set in a pattern sort of at a pretty early age, right? So like I think I've read like 3 to 5 years old, your microbiome is pretty set. And, all of that comes with with birth, the species that you have, and they can change and they can shift, but the colonization of the species is pretty set early on. So are you
talking about and how they react to certain foods and stuff? I guess I don't really understand what's happening.
Like your pattern, like if they did a genetic sequencing of your microbiome, you'd have around the same rough pattern throughout most of your life, besides like big disruptions like antibiotics and stuff like that.
So like the healthy baseline began.
It's like a fingerprint, like your fingerprint's done, but it may slightly change, but the fingerprint's there.
Gotcha. So that's what we're aiming for when it comes to our healthy biome.
Well, I think we're always aiming to improve our gut microbiome, and we're always wanting to change things and to improve it. But the pattern kind of starts from birth, is whenever you're given your microbiome is sterile whenever you're in the womb, and then the birthing process is whenever you get inoculated with your first part of your microbiome.
Okay.
And then it changes and shifts for a while pretty rapidly until you get to, I don't remember the exact age, but somewhere around like 3 to 5 years old. And then at that point, you kind of have, like you said, your fingerprint of your microbiome, and it can shift and it can change a little bit, but that's kind of what you have set.
Like an ecosystem. Like the ecosystem's formed, and if you start affecting it, it'll have to.
Right. Well, to go back to the zoo analogy,
Yeah.
like the animals that you have in the zoo are mostly going to be set at an early age. And so we're always either at that point, things are going to be either dying off or we're going to be trying to get more diversity in there. So I guess my question is that how your understanding that we start pretty early on, like what we have in our pattern is set pretty early? And then
and so they've had a lot of data on that where, obviously, like you said, the GI tract of, before they're born, it's sterile, colonized by mom, or if it's a C-section, colonized a lot of times by the skin flora of the nursing staff or whoever's interacting with the baby. And then, a lot of that will change over those first few years, especially like if a kid's raised on a farm or if they have a pet that's, licking them on the face or going out and getting a lot of different exposure. But the microbiome is very much like a fingerprint where it's going to be very different amongst individuals. Even if, say, you have, triplets that live in the same house, they were vaginal birth, which they're triplets, it might have probably been a C-section, but they have a pet in the house, they have all the same exposures, and you did genetic sequencing on the microbiome, they're not going to be the same. They're going to be individualized for each of those individuals. But they still may have more diversity based on what happened to them, like if they didn't get antibiotics or if they were raised with a pet. So a lot of it you can't, we don't know, and we're very far from knowing like what is the fingerprint microbiome that you would want. And even if we thought we knew, it's still going to be different based on each individual. But yes, it is very specific, and it's very much akin to a fingerprint, like, from that first few years. And then what you do after that is going to greatly affect the diversity and the balance of the ecosystem throughout your life.
Let's talk about the disruptors. Like, what is the biggest disruptor to your microbiome, or some of them? I mean, you mentioned antibiotics.
Yeah, antibiotics, especially the way that antibiotics are designed by pharmaceutical companies, they're not typically designed to kill one microbiome, one microbe. They're meant to kill, several different organisms, which it's beneficial for the pharmaceutical company because you can use that drug for urinary tract infection, for pneumonia. You can use it for several different infections. The bad part is it indiscriminately kills, a large chunk of the microbiome. A lot of times, even when patients get diarrhea after taking antibiotics, it's just a mass bacteria kill off. The problem is, with that situation, you don't know what's going to reconstitute the microbiome. Some of your baseline species are going to stay, but you don't know if something pathogenic like Candida or a parasite's going to sneak in that window where your microbiome is not back up to full speed.
I'm sorry, did you say Canada?
Candida.
Oh, okay.
Yeah, not Canada.
If I go to Canada, what?
Canada moves in, we've got way serious
problems. Yeah. But in addition to antibiotics, things like stress, things like poor diet, like high sugar diets, metabolic dysfunction can affect the gut microbiome, where you have issues where, you're having these very high blood sugars and you get chronic inflammation in the cells and mitochondrial dysfunction, which can also lead to things like leaky gut and can also affect the microbiome. But if I had to pick some of the top ones, it would be poor diet and antibiotic use would probably be the top two. And then you kind of sprinkle in like, poor sleep and lack of movement. But everything affects the microbiome. So the things that you do for good health for your own body are a lot of the things that your gut microbiome has developed over the years as what's good for them. Because these things have been with us for thousands of years. So the things that we used to do, like move a lot, eat lots of fresh fruits and grains and high fiber diets, sleep adequately and avoiding toxins and pollutants. And obviously there were no antibiotics thousands of years ago. So the microbiomes developed in that situation. And then all the things that we've changed and added, especially antibiotics, significantly affect their populations, decrease the diversity, and then, like I said, open the door that something else can come in whenever those populations are low or affected.
Sounds like we all have wrecked microbiomes then, because these are things that we're all experiencing. Like, we all are eating the American diet. We're all exposed to pesticides.
Yeah, I think a lot came out of the Gut Microbiome Project, where they recognized that we all could use a little help, and to be able to say that something is a healthy gut microbiome is just kind of an arbitrary, like, we don't really know exactly what that looks like. I think all of this is all still very new, like where it's in its infancy of understanding the interactions that happen between microbes and how that interacts and affects the way that the body functions too.
You said you can test it? Like I can get a test to see.
You can sequence the whole genome.
What does that, like you said, but you also said there's no
blueprint for the perfect genome. Yeah. Like if you tested yours and they were like, is this good? And it would be more looking at how many species do you have? Like say you'd want 900 or 1,000 different species to have lots of diversity versus they tested it. You had 400 species and you were lacking some of the key species that they look for.
So there are some that we know
that are beneficial. But even then, some of those ones were like Akkermansia muciniphila is a good example of one where it's great if you have it, but a lot of people they won't get colonized with it.
I do not.
My daughter has zero traceable, detectable.
No detectable.
Yeah. And so it's like, we know like, oh, that's a good one. And we say it's good, but then if you look at all the biochemical processes that are happening, what the person eats, their hormone level, their age, how much sun exposure, like if you look at all those variables, it might be like, well, it's not really that important for that person, so it's like, we're like fourth graders in our knowledge of what species are great.
We just know the more you have, the better, because it can.
Exactly. You have more diversity. So maybe a lot of them are doing not much of anything, but then those, 300 or 400 species might be doing something great. So the more diversity you have, you kind of have organisms to fill in those gaps.
Gotcha. Okay. That's cool.
They'll just compensate for one another as needed.
Yeah. We did the GI map testing, which I'm very curious to know how the genome mapping, is that what you call it? Genome testing and the GI map, are those two different tests?
Well, the GI map's looking at quantitative PCR for different bacteria and parasites, and so they kind of set out a blueprint where like, oh, if you have Pseudomonas or if you have this, and depending on what concentration of it, it may be pathogenic, or if you have lower numbers of this, it may suggest chronic inflammation. So they've tried to build like almost like a template of different things where you're looking at proportions of certain key bacteria, but they're not showing you all like 900 species.
Right. Yeah. It was how many? I don't remember how many show up on that.
I don't know off the top of my head.
It's not 900.
No, it's not 900.
But my daughter and I did the GI map at the same time, and she had no detectable traces of Akkermansia. And in your opinion, does that matter? Is that something that needs to be treated, or is it, because I know that's such a new one that we're starting to talk about?
It's so tough because it's like when you really start peeling back a lot of these things, you start to realize, a lot of it, not that we're guessing, but we're trying to make an educated proposition that, if I have this bacteria that's associated with better metabolic health, then testing people for it and then potentially trying to put it into capsules. There's one company called Pendulum Therapeutics that has some different ones circling around metabolic health, but they're using Akkermansia muciniphila. But the problem is when you stop taking it, a lot of people will lose that bacteria. Off. And when you're looking at the scale of life and you want to do something that ideally you're going to do for 40, 50, 60 years, it's very difficult for somebody to say, I'm going to take a probiotic for the next 40 years every day. So it's
also not cheap.
Yeah, you start getting into a situation where you're like, what are we doing? And so, a lot of the pioneers, and especially the ones that have gone far down this road, unless they're developing supplements for people to buy and take, they're just pushing overall health and doing the overall healthy things and avoiding the things that are detrimental. Because when you start trying to select certain species for certain people, it gets dicey, because I'll give it to you, you get bloating. I'll give it to someone else, they feel great. Give it to someone else, they lose weight. So you kind of get into this rabbit hole of like, what are we doing? And is it going to be something that we're going to do long term? And it's great that they're studying and looking at all this, but to some degree, I feel like we're grasping at straws, because it would be easy to just take a probiotic where you're like, oh, it fixed my metabolic health. Insulin resistance is gone. Now I'm colonized. I'm going to feed this thing some broccoli a few times a week. Everything's good. Where in reality, you're like, you don't even know the role that bacteria specifically would play for you, and then how it fits into that system of 800 other bacteria species you have. And then if you're going to keep doing it, and then if you're going to eat the certain foods that can maintain it and let it work like it's supposed to. So I don't say all that to say we should give up on that type of stuff. I say just use caution when trying to to select certain bacteria for a very specific goal, because just looking at the big picture, there's a lot of other high yield things that you can do without just having to take a supplement and spend money on it.
Right.
Well, and then if we're talking about what, thousands, hundreds, almost about 1,000 different species, possibly, what % of those do we not know what they do or why they're there?
Significant portion.
So a lot of those, if you have them and don't know what they do, but you're trying to get another one by supplements or eating a certain thing, you may already have one that does that. We just don't know it does that.
Exactly.
So it's like pointless to begin with.
So many of those bacteria can't be studied because can stay alive in an anaerobic space where they don't have oxygen. So we obviously can't study that if we're bringing it into an oxygenated world, then we can't study those bacteria. So I'm interested to know, what do you think about...
But not all of them? So some of them can be...
Mm-hmm.
Oh, okay.
Yeah.
Gotcha.
What do you think about just probiotics in general? Is there a place for someone who comes into your office and they're having gut discomfort, or they just want to preserve their health? Do you recommend probiotics often?
I do for certain conditions. So there's a lot of good data on IBS, constipation, and IBS diarrhea. I always tell this to patients, it's not like, you have a disease and we have a treatment that's been proven that it's going to work for this disease 100%. The tough part with probiotics is because it is such a complex environment, I'm very much a proponent of, we can try it, but you have to listen to your body. So for example, if somebody comes in and they feel great, but they're like, do I need to take a probiotic? A lot of times I'm like, no. If you feel great, let's focus on the fermented foods, fiber, and all the other things that you do for health, like sleep and exercise. If somebody comes in and they're like, ever since I'm 17, I'm 40 now, I've been having intermittent bowel movements, a lot of urgency, and they kind of paint a picture of irritable bowel syndrome, and say you've done all the testing, that's what they have. You checked them for parasites, you've done all the appropriate workup. Those are the patients where I like to try the most diverse probiotic you can find, like something like Seed or one of these companies that have a multitude of strains of live bacteria in good proportions. And then I'll say, let's give it, a 2 or 3 month trial. We kind of start slow, work it up. But then if at the 3 month mark, they're like, I notice absolutely no difference, I'm also a proponent of saying we need to try something different, because it's not the end all be all, because we know those reactions and those relationships are so complicated. I don't want to add it to a regimen just because, but then some patients take it and they like, I feel great. What's the end point? And I'm like, that's a great question, because a lot of these bacteria don't colonize or don't stay. So I'll say, look, let's do 6 months total, and then we'll stop and we'll kind of slowly wean off of it and see what happens. If all your symptoms return, then at least now it's a good enough reason to invest in this every month and take the powder or the pills or whatever you have to do. Versus if they stop it and they're like, I feel great, can I just stay off of it? And I'm like, of course you can. Yeah. So I approach it more in that manner.
So it's all just kind of trial and error.
Yeah, not a yes, no, more of a gray area it's another tool in the arsenal.
Yeah.
So last week I went to a talk by Dr. Christopher Lowry, and he's not against probiotics. I think he might even have a portion of a company that creates them, but he was talking about the variety of plants that you eat and how that affects the microbiome. And apparently there was a study recently, was it the number of plants you eat in a week we had talked about, that affects the population, and people that ate 30 plus plants a week had the best microbiome. So now he makes this little concoction of plants, and he's a neuroscientist and he studies how the gut affects the mind, but he's been doing this for years. He's done a TED Talk on it. Do you talk to your patients about that, like implementing a variety of plant sources?
Yes, and I'm a big proponent of, especially that patient like I just described with IBS diarrhea or something like that. I like to try to get them to do the paleo diet. I'm not a big fan of the term diet, and Daphne's a dietitian. She probably has a lot of the same ideas, where it's like you want to push them towards certain food groups. And I don't like to use the term diet because I don't want them to feel like it's something that they're going to do for a period of time and then stop.
It's like a lifestyle.
I want it to be, this is a life change. And then you can slowly reintroduce stuff that you might want to, if it works out great, you can reintroduce stuff slowly. But I'm a big proponent of different in variety of vegetables every single day. Not just a little bit, with a little bit of a meal, like half the plate is vegetables, the other half is the other stuff, the meat or the carbohydrates. I'm also a big believer that there's no specific diet for anyone. I'm sure Daphne has tons of data on this too, but there's very different diets for very different people, but there's not many of them that don't need fiber, especially when you're trying to hit a goal of, 30 to 40 grams of fiber a day. Good to have lots of variety. So I always say, like, load the plate up with lots of different colors. So, oranges and reds and greens, get in cruciferous vegetables like Brussels sprouts and cauliflower and broccoli, and then doing fermented foods too. But I'm a huge believer I've seen more patients get better with doing diets like the paleo diet and upping their fiber intake gradually and lots of fresh fruits and vegetables than any medicine or probiotic that I've done for IBS diarrhea. And the good part is you have to eat anyway, and it's going to improve, your insulin resistance, your diabetes, your high blood pressure, your high cholesterol. So it's nice because one intervention is doing multiple things.
Is there anything specific about the paleo diet structure that pushes your people in that direction?
I like it because it cuts out a lot of the autoimmune reactive type foods, especially the main one is gluten that a lot of people obviously have issues with. I also just include seed oils because I feel the same way with that. And I feel like it makes it a little simpler because I'm like, you can eat all fruits and vegetables, lean meats and fish. And I'm like, it makes it easier as a starting point. And then if they come like months later and they're like, oh, I want to, I really liked wheat bread or whatever. And when I eat it, I'm fine. Then we can kind of like dial back some of that stuff. But I feel like it's an easy starting point, especially in South Louisiana, because a lot of people have gardens. Mm-hmm. A lot of people already eat lots of, fish and seafood. I mean, you don't have to twist arms to eat meat in Louisiana either. So it just feels like an easy starting point for a lot of patients.
So the gluten-free part you feel like is an important aspect of it, though?
I do. Not to go too much into a side note on gluten allergy and gluten sensitivity.
Do it.
But so when you think about the immune response to food, so you have your traditional true food allergies, which are usually IgE mediated. So that's something where, and they actually even think with food allergies that it begins as something with a skin disruption. You get exposed to, like, say, peanut dust. It gets in your skin. Your immune system reacts to it, where you have these cells called macrophages come in. They take in the peanut particle or protein, to be more accurate. Then they present it to T cells, and then the T cells bring it to another cell called a B cell, which now produces an antibody against the peanut particle. We know that's how the allergy system progresses. Sometimes it may have been a food exposure, but a lot of times we think it was skin or somewhere else. Then now, anytime you have that food, whether it be, wheat or milk protein, or for celiac disease, gliadin protein, now you have antibodies that come, and then you get these mast cells that just dump a bunch of histamine, and you get, rashes, itching, nausea, vomiting. You get a severe reaction.
Do the eosinophil cells ever come into play in this part?
Yeah. So mast cells are in the tissue. Eosinophils are in the blood. Same function.
Okay.
And basophils too, but eosinophils and basophils are cells in the blood, whereas the mast cells are in tissue. But they're both binding with the immune globulins and releasing histamine.
Okay, because I have eosinophilic esophagitis.
Same spectrum.
That's what I'm wondering. Okay,
yeah, same spectrum.
Cool.
That's the pathogenesis of a true food allergy, which for celiac, for gluten allergy, it's probably about 1% of the population, maybe a little more, a little less, depending on gender and race. But for gluten sensitivity, which is more reacting to a small molecule, like a sugar molecule or an additive that's placed in the food, is much more common. And this is just me guessing, but probably about 30-40% of patients will get improvement in symptoms if they cut out wheat and dairy. Dairy, a lot of it is lactose intolerance, which constitutes a food sensitivity, not a food allergy, because they're reacting to the sugar in milk. But it's very important to kind of start with those big two, and then I just kind of work it from there. Which milk and wheat, especially in the United States, is a more common one. And for example, in other countries like in China, fish is a more common true allergen than peanuts here. So it's like every country kind of has their thing. And it's probably because what's very common in the environment whenever that child is young and gets the exposure and goes through that pathway.
When you say the sugar in milk or the sugar in the wheat, what do you mean by that?
So when you have a true allergy, it's typically reacting to a protein.
The protein, right.
Whenever you're having a sensitivity, it's usually a different reaction pathway, and it's usually a sugar molecule, like lactase for milk, because you don't have the enzyme to break it down. So it's not an allergy, it's more of a intolerance because there's something happening with the breakdown of it. Okay. Either it stays in your GI tract and it gets fermented and produces a lot of gas and bloating, or you're having like a more mild reaction to it. But it's not the histamine pathway.
What changed in the past 30 years, 20 years that all of a sudden everybody's allergic or sensitive?
Well, we know there's a lot more allergies, and so it plays into a huge picture. And so, you have the changes in your microbiome, which are definitely very important in playing a part in identifying like things that are good or bad for the body. So the microbiome is getting exposed to all the same foods that the GI tract lining is getting exposed to. And so it plays an important role in keeping a good barrier to where, if chicken protein comes in or egg protein comes in or wheat protein comes in, it helps the body identify this is safe. You can eat it. You don't have to make antibodies to it to react to it later. So that part has changed because, there's a lot more C-sections, which is good because it saved a lot of women who would have died potentially during childbirth. But it changes that dynamic. And then you also have a lot of push, years ago by pediatricians to not expose kids to certain things by a certain age. A lot of it was eggs and peanuts, which that's why you saw a huge spike in nut allergies. And some of that language is quietly being dialed back. It's not shouted from the rooftops like whenever they, really pushed it out there not to expose kids before 2 or 3. And then in addition, industrialized society, like kids are not out playing in good dirt. And so not dirt filled with pesticide and runoff from the roof with tar and all that stuff. And a lot more families had pets and farm animals and their own gardens. And so a lot of the immune response is that the body hasn't been exposed to a lot of stuff where it could have said, oh, this is good for me. This is fine. Let me not have a reaction to it. Versus now, the body sees some stuff for the first time because we're just not really exposed to it in those first few years. Babies are kind of raised almost sterile, like you wash your hands.
Don't touch the baby.
Family comes over, which my wife and I were, huge for that. Like, don't touch them. You're kind of sniffling or whatever. Don't let the dog lick the baby. You drop the pacifier, you like disinfect it. And so, a lot of that.
I was sharing a pacifier with like my aunt's dog.
And so it's like, when you take that spectrum, a lot of those early exposures were very important for the body to start to develop a sense of, safe or not safe to make those reactions happen. And so you see this huge jump in reactions. And then also the way that they've refined and changed wheat protein and all the ways they strip it of a lot of its beneficial parts to make the shelf life longer. That also plays a part in why people react to it so much more. But we're seeing more food allergies and sensitivities across the board. But then all these foods have also been altered and changed and covered in pesticides.
The way that wheat has been grown over the past 40 or 50 years has changed, where it used to be long and tall, and now in order to process the crop faster, it's been hybridized. So not necessarily genetically modified, but hybridized to make the wheat plant shorter, and the chromosomes of the wheat itself there's much more in it. It's more consolidated. So it's completely changed the plant, the end product of the plant, by the way it's been hybridized.
I've heard a lot of people say, like, obviously not celiac, but just gluten sensitivity, they don't have the same reaction to wheat from other countries. American wheat is the one that affects them the most.
And so some of that plays into a sensitivity where you have in these other small molecules that the food processing industry in the United States is just altering it to a point where you're reacting to all these other little things in it, not the actual, wheat plant in its truest form. And so that's why people will go to other countries and be fine and then come here and having issues with it, because a lot of it is you look at the ingredients. Even if you went and had pizza at a restaurant where you thought it was, naturally made pizza, and it's like 40 different things. So you could be reacting to any of those.
Yeah.
Professional opinion. What's the line of throw my baby in a dump truck to keep my baby in an incubator?
No, that's a good question. And, a lot of times, unfortunately, like the pendulum will swing the opposite way. And it's like some of these things like antibiotics and sanitization have saved, millions of lives, especially the invention of, the discovery of penicillin was one of the biggest lifesavers, that any of us have known about. But there is a spectrum. So it's like, what we say is, and we kind of do that for our kids, we call it, good dirt versus bad dirt, good exposure versus bad exposure. So if your kid goes to, say, Surge, and, they have multiple kids with stuffy noses, and I'm sure some kids got a viral GI bug, and you're in that environment with 100 kids,
bad dirt,
hand sanitizer, wash hands, like shower, change your socks, your clothes. But then if you take your kids, like we go to my parents' house, and they live out in the country, and they have several acres. There's no houses close by. No one's spraying pesticide on crops or anything. And so the kids get all dirty. Like they just come in, eat lunch. Like sometimes they wash their hands, sometimes they don't. Same thing with our pets. Like we're letting the kids like kiss on the dog, play with the dog. But then if we bring the dog to a dog park and it's interacting with 20 other dogs that one of them probably has Giardia or two of them, then we like, for a few days, like don't kiss the dog, don't hold the dog. So there's a lot of, trying to tease out, what's a good healthy exposure, because with that micro environment that you have, you want to introduce good things. You don't want to introduce bad things. So you don't want pesticides, pollution, and pathogenic stuff like Giardia and E. coli and stuff. So you want exposure, but you want to just be mindful of it.
Be smart and cautious, but not keep them in a tube.
Yeah, not a bubble.
Yeah. Don't lick the floor at Surge.
Yeah.
But you can lick the floor at
lick
home.
The grass at the farm.
Yeah.
Something that we talked about at the Health Revolution was 70%, and I don't know if that's the exact number, but 70% of your immune system is in your gut. Can you talk to us about what that means?
The GI tract is very interesting. One of it is because it has so much real estate. So you have over 20 feet, and then you have structures like villi, which increase the surface area of the GI tract, but you have so much lymphatic tissue and immune system tissue there, which makes sense because everything that's going in through your mouth is gonna get exposed in the GI tract. So it's a big barrier where you want a good defense system for parasites and viruses and things that might cause your body harm. And then it's also an important part of the body where you're going to get a lot of exposure, where you want to start to learn, safe or not safe, because the skin is different, where the skin is more of a barrier where things are not coming in or out, and there's not a lot of communication with the skin and the outside world. So it's really inside the GI tract where most of the communication and the bidirectional interaction is going to happen. So that's why there's so much immune tissue there, because it has a big role, not only in protecting you, but also teaching the body safe versus not safe, which is where you see these derangements with allergies, inflammatory bowel disease like ulcerative colitis and Crohn's, where when things go wrong, because it is such a big immune system target that's there, you have a lot of things that can go right or wrong.
That's pretty much the driver of autoimmunity too, is what's happening in the GI tract.
Completely.
Right? Yeah. So let's talk about H. pylori. I'm like fascinated with H. pylori right now.
What's that?
So it's called Helicobacter pylori. It's a bacteria that lives in the stomach, the actual stomach. Certainly you know more about this than I do, but my understanding is that H. pylori, about 50% of the people of the population have it. And some of those have challenges or issues because of H. pylori. But it's the reason that it can become a problem is that it slows down the gastric acid production. And then when that happens, it slows down your digestive enzymes that are being produced by the pancreas, but also keeps your stomach kind of open for bacteria that come in through your mouth that should be killed by the gastric acid, but are not necessarily killed by it. And so it can then pass through and become a little bit more pathogenic further down the pike. What is your stance on H. pylori? Some people would treat every bit of H. pylori. Sometimes they'll talk about only treating it if it's problematic. if that's the case. I mean, we could go down and have lots of questions about H. pylori, but also how is it transmitted? There are just so many things that we could talk about.
Yeah, so H. pylori, so there's a lot of different points about H. pylori. I'll kind of start with earlier in life, and this was an actually interesting thing I read about, several months ago, but having H. pylori, especially like in teens and early childhood life, significantly decrease the risk of childhood asthma and adult asthma,
with
and a
H.
lot of
pylori present?
allergy issues with H. pylori present.
Okay.
And so, there was...
Is this a type of bacteria?
Mm-hmm.
Okay, that's found in the gut. Okay.
And it's found in the stomach specifically.
Gotcha.
It kind of goes back to what we talked about before, where there's big pushes for, yes or no this. And then you get this organism where we don't know, does it play a role? Because like we said before, these organisms have been in our bodies for as long as we've been here. So, yes, they do have negative effects, but they're very person specific. And then there also always has to be the question, could they have a beneficial effect? And so one is in that early childhood time, whenever you have H. pylori positivity, you have less chance of asthma later in life and less issues with allergies. We don't completely understand the role. It's more of a correlation where we see, kids that are positive for H. pylori have less of these things than kids that are negative for it. And it could play a role in like their environment and what they're exposed to. Do they have pets? All the stuff we had talked about before. But then you fast forward into adults. So H. pylori can do a lot of different things, and a lot of it depends on different virulence factors and different genes that different H. pylori bacteria will have. So some of them will have virulence genes that can lead down the pathway of gastric lymphoma. Some of them can lead down a pathway where they're actually protective against esophageal cancer if you do have it. And then some can lead down a pathway where they increase your risk of stomach and duodenal ulcers, especially in combination with NSAIDs like ibuprofen, Advil. And so you kind of have a spectrum where in some instances they're good, sometimes they're bad. And then depending on where you have the infection in the stomach, like if it's antrum predominant, which is this part of the stomach where it attaches to the small bowel, you'll actually get acid hypersecretion because it's affecting the cells that normally help downregulate acid production. So a lot of times we think in that acute phase when you first get H. pylori, you'll have an acid hypersecretion, which may be that window where more patients get peptic ulcer disease, which is just speculation. It's impossible to know when they actually get it. But then we know the bacteria tends to migrate into the stomach body, and then it starts affecting the cells that actually produce acid, and then you get a lower acid production and you get more of a basic environment inside the stomach, which that increases your risk of things like C. diff and pneumonia because you need that stomach acidity, not only for digestion, but just to protect you from stuff. And so, it's funny, just when we think we have things figured out where across the board we were treating everybody for H. pylori, you get all this data and you start getting gene sequencing and you start seeing the role that these bacteria play. And now you find out even one that you thought, oh, it's a no-brainer. You find it, you treat it. And now, you have to be more of a steward of looking at the patient as a whole and what's the overall context. For me personally, if a patient comes in and say they're coming in because they had, we suspect blood in the stool, we do an endoscopy, which is, where we bring them in, they get sedated, we take a look inside their stomach. We take samples for H. pylori pretty routinely, especially if there's inflammation or ulcers, and say that patient has an ulcer and they have H. pylori, we treat it. Because, the risk of potentially bleeding more from the ulcer is just so high, we do it. Then you have those patients that are, non-specific symptoms. You do the endoscopy, you take biopsies of the stomach. It's a little red, but there's no real ulcerations. It's not really severe. And then you get H. pylori. And then you call the patient and they say, I'm allergic to all of the medicines that you use to treat H. pylori. So now the question comes, do you send them to an immunologist to see if they're truly allergic to those antibiotics because you really have to treat the H. pylori? Or do you say they don't really have, any of the disease present that I have to treat it? So now I think we're getting to that point where we're going to start testing for those virulence factors in the H. pylori to see, is it one that might progress you to gastric lymphoma, or is it more of a benign strain that we can just say, hey, let's just leave it alone? So it's definitely patient specific. But for the most part, more patients get treated than not. But then sometimes if you really want to treat it and they're that high risk patient, you have to treat the spouse and test them.
Yeah, that
because
was
then
my
you
next
can
question.
re-catch the infection.
Yeah.
So it's a tough to treat infection. About 20-30% of the time, you don't eradicate it with the first round of antibiotics. And it's a tough regimen. It's several antibiotics over a 2-week period. Most people don't tolerate it well. So then you have to do another regimen, which is also not tolerated well. So it's definitely think in your mind, like, would this benefit them if I treated the infection, or is this going to just make them sick and I'm just doing it because?
And those antibiotics are causing other problems, I'm sure.
Exactly. And then you loop back around to like, what are we doing to the microbiome? What are we doing to the system? Like, you're going to burn down the rainforest because, there's one thing you're trying to get rid of when that thing was maybe beneficial.
This is an infection?
This is, well, you could call it a colonization more, because
That's what I'm
some
wondering.
people have it for years.
And it doesn't affect.
And some people, it doesn't do anything.
This is where my mindset goes. I have H. pylori and I don't have no virulence factors. My digestive enzyme production is low. And so my assumption is that that's coming from the H. pylori. And then I also have autoimmune disease. So in my mind, the H. pylori is causing the digestive enzyme production to be low, which is causing proteins to not be well broken down, which is kind of going into the autoimmunity. So that's just my quick thought process. So that's kind of why I was asking, is because I know there's a lot of factors that happen, a lot of things involved. And
but is it still all just correlation, or is it, do we know it's causation?
Some of those virulence factors, we know they'll lead down the pathway of lymphoma, or they'll increase your risk of gastric cancer.
Okay.
Whereas my husband had also H. pylori with virulence factors, and he was treated with antibiotics, and he's got all kind of gut stuff happening. And finally, he's let me be a part of helping him.
Yeah.
And once he was treated, he's like, I feel amazing. I feel fabulous. So much better. And so it just makes me wonder, like, what's the line of where is it treated? What, if you suspect that it's causing a downstream effect, like, then it seems like it would need to be treated, but there's not always the question of, is it problematic or is it not problematic?
You mean he was treated for this specifically?
Mm-hmm. For the H. pylori infection or colonization.
You could probably just, and you probably did this already, just supplement with, apple cider vinegar, try to like drop the pH a little bit to help supplement and see if it changes.
Is that like,
sometimes if it's like it'll just make it more acidic.
But like,
so like the H. pylori, what she's talking about, if it affects the stomach body itself, it's decreasing acid production. And so sometimes, which is only about 15% of the population where they don't have enough stomach acid, because the pH is supposed to be below 4. And so like, say theirs is 4.6, which makes a difference physiologically. Maybe you can just try to drop it down a little bit with, apple cider vinegar, stuff like that. And then if you're like, oh, this is great, after like 2 or 3 weeks, and then probably just treat it. You could catch it again anyway, Well,
that's the other thing. Like, so do I need to test my son? Can I never, ever, ever drink after someone again? Like, just how far
part.
Does it go down? Like, somebody's like, hey, taste this. Should I be like, oh no, I don't want to get H. pylori possibly from you? It's like, how far do we go down this path?
The prevalence is so high though. It's like, to me, you just treat it, and then you can always do the stool test. Like, if you're like, man, I think I got it again, like a couple of weeks after, just take the stool test,
So it can go away and not come back, but you can get it back.
Yeah, because the prevalence, like you said, is almost half the population. So like, you're probably going to get it again. But if it doesn't cause any issues, I never check for it again. I have no problems. We just kind of just let it lie.
So you're saying if your stomach's too acidic, drink the apple cider vinegar?
No, not acidic enough.
Not acidic enough, add. Okay. So if mine was...
Because you need it to be a certain pH to do certain things with starting the B12 absorption process from meat. Like, a lot of things need the acidic pH. It needs to be low pH and acidic.
I remember looking that up because GERD also have that. And so I just wanted to try high pH water, and it felt like it helped. But I didn't know if it was actually just like a mind thing.
Well,
or if
the
it was
high pH
me.
water helps wash the esophagus, but saliva is supposed to kind of do the same thing because you have bicarbonate in it, but it won't hurt.
So it's not, it wouldn't like lower the...
Especially if you did it after, like you went to eat Mexican food and like 20 minutes after you had an alkaline
like just
water.
rinse it out?
Yeah.
Okay,
But your saliva is doing the same thing all the time.
gotcha. That's cool.
I did a GI map test with Daphne, and there was a bunch of stuff on there, which actually triggered me going to see you. And one of the things, I think I had H. pylori on my GI map, but whenever I did my endoscopy and colonoscopy, H. pylori was not on there at all. So
yeah, so
that's weird.
So even the GI map test. So remember, you're trying to pick up a bacteria that's inside the stomach, and you're trying to get it in a stool sample. So it has to pass through all this area. And the H. pylori bacteria lives and thrives in an acidic environment. So if the environment gets pushed too basic, so you push the pH up too much with like PPI therapies and stuff, sometimes like the bacteria almost go to an undetectable level. And then for other people, the bacteria is in such a low level and it's so sparse in the stomach that you may pick it up with a PCR stool test, because that's, amplifying the DNA of those organisms, and it's super sensitive. Versus if you're taking biopsies, yeah, maybe if we took 40 biopsies, we might have picked it up in the stomach. But sometimes, you still have a false negative rate where if you don't take a ton of samples, you might miss it. But my kind of thought process with that too is we take adequate samples. If it's not there, it may still be there, but if it doesn't look like some condition that's attributed to H. pylori, exactly. Because then we get into the conundrum of, I took 20 samples, stomach looks great, besides all the bleeding from all the biopsies I took, and then it's positive. What do you want to do? And then it's like, someone's like, I don't want to take Flagyl or this antibiotic. I just got my microbiome like doing well. I'm eating fiber. And we're like, oh, well, it's positive now. So, so I take adequate samples, especially if I don't have a high suspicion of a H. pylori related disease. And then if it's negative, you may still catch it in a test like this, but I don't think it's as big of a factor.
So the reason why I did a colonoscopy with you is because I did this GI map test, and there were so many indicators that I needed to do that. And I was 35 at the time. And I don't know is it 45 where they start recommending?
It's 45, yeah.
And they just lowered that.
Big risk factors. Yeah, it was 50 before.
Are you seeing an increase in rates of issues? Because I know like in the United States, colon problems are increasing. Are we seeing that here?
Yeah, we're seeing an increased incidence every year with patients, especially the patients under 50. And the biggest thing is the surprising part is patients like with you, we go in and we find a polyp whenever someone's in their 30s, and we're like, if you'd have waited 10 years, especially, the type of polyp where it's over a centimeter, it has high risk features, the chance of that progressing to cancer is pretty high. And the terrible part about that is it's the one cancer where you have a clear progression where you get a polyp, you have a visible lesion where you can see it, like I can see the polyp, and the tissue's normal, and then it progresses to, it's a little bit more abnormal, and it gets bigger, and we can still see it, and then it progresses to cancer. And the scary part is this happens over years, and so it feels like such a disservice if you get somebody who comes in at 52 and they have a colon cancer, because you're like, we had such a big window where we could have found it and prevented it. And especially, anybody who's interested in longevity and increasing their health span, in addition to their lifespan, like people that are big proponents, like Peter Attia, like he got his at 40. And he's done several, and he does it every 5 years, even though it's normal, which for us, if it's normal, we recommend 10 years because that's the guideline, even though we don't feel great about it, because of insurance paying for the procedure and stuff like that. But, if you could just get patients to come in even at 35 or 40, get one done, and then the baseline, because these polyps are not just appearing out of nowhere. They start off and, they might grow maybe a millimeter or two a year, depending on the type of polyp it is. So it's like a very slow train wreck.
Can you describe like what a polyp is, and if you would develop a polyp, does it ever go away without removal? As
far as what you'll see to the naked eye, it's just, just like you would imagine, like if you saw a skin mole, except it was the same color as your skin. It's just going to be a raised
just a formation
area
of
on
tissue.
Just a raised area of tissue. But the cells have what we call low-grade dysplasia, which is just a term to say they're not normal, they're not cancer, but they're kind of something, in between. Closer to the normal side, where most polyps that you develop are not going to turn into colon cancer. Probably 90% plus polyps will never turn into anything. And then there's different estimates, but, maybe 5 to 6% of those polyps, if they get big enough, can make that leap into cancer. And so the problem with the colon, unlike your skin, like if you saw something on your skin growing and changing and you're like, oh, something's not quite right, you'd go and you get it taken off. Whereas the colon, if you don't get one done till 58, it's 58 years where something could have been in there, and time is just not on our side. So having polyps is very common. For everybody, it's different because we have different detection rates. Some doctors are very good at finding polyps if you have them. But, in people with a very good detection rate, it's an upward of 50% of people will have a polyp. So you're thinking half the people have a polyp, we should be doing this much earlier. It's not like 5% of people have polyps and we're being over exaggerating. It's like when half the people or more have polyps, I want to look at 40 or 35, just because you can also address other symptoms like for your situation, but you find that polyp, you take it out, you prevented colon cancer.
So once a polyp develops, it doesn't go away. It's not going to just you can't change diet and make your polyp go away, as far as we know.
Like it doesn't heal.
No.
Okay.
No.
So a couple things. I was 35. After I got my colonoscopy, I felt so much better. I guess it probably was a little bit to do with the cleanse that I had to do,
that's exactly.
which was not fun, but very effective. And also removing the polyp that you found. My life was so much better after that came out. I've told everyone, you need to go get a colonoscopy.
Yeah.
I bet mentally you felt better too.
Well, something that we don't talk about in polite society is how our poop is and what's going on in our stomach. And it's kind of taboo and weird to discuss that. But if you're having these issues that are embarrassing, probably something that you need to talk about. Your poop's supposed to be what's that graph where the different types of poop?
The stool chart, the Bristol stool.
Yeah, the Bristol stool scale.
I love that thing. And my daughter has GI issues. I have GI issues. And now I'm like, okay, what number is your poop? You're like, what does your poop look like? And feel like we need to start talking about that more, because we are having we all have GI issues.
Yeah. There was a great saying
everybody
saw,
poops. Is that the saying you're about?
Yeah. But there's a great saying, and I'm forgetting who said it, but they said in a society, if the poops are small, the hospitals are big, and if the poops are big, the hospitals are small. It's a great analogy because you see so many issues developing from just poor bowel habits, poor fiber intake. And it just correlates with so much other stuff. And it's definitely a spectrum. People that are eating the fiber goal are probably also exercising, meeting the water goal, meeting the sleep goal. But definitely, it'd be so awesome if people would just start somewhere, especially like you did. You just come to someone and you bring the problem, because sometimes you just suffer in silence and you don't say anything. And, you're like, am I the only one? And then when you come, the first thing I always like to tell people is, we hear this all the time, not to say it in a way like, I don't want to hear it anymore. You're not alone. The doctors included, the nurses in the practice included. We all see each other. We all get our scopes at 42. Not 42, 40 also. But it's good to know you're not alone and there's ways you can get better.
Yeah. There were so many symptoms that I had that I didn't realize were correlated, that I thought I was just weird. This is so embarrassing, but I'm just going to say it because we're talking about poop. My husband would always tell me, for someone so cute, you smell your poop smells so bad. And it was mind blowing to him. And that was a sign that I was having issues. Your bowels aren't supposed to smell that bad. Obviously poop doesn't smell good, but it was an indicator that something was going on in my stomach. And my polyp was 3 centimeters.
Yeah, yours was big.
That tells me it was in there a long time ago
to turn into cancer, especially by 50.
Yeah. But if you're saying they grow 1 millimeter a year, does that mean it's been in my body forever?
Well, and then the one you had specifically, there's different features of the polyps that make them higher risk to progress, grow faster. And so the size over 1 centimeter was one, but it had villus features, which is another risk factor where it would have gone further and been higher chance of cancer than the 5-10%. We don't have a data to quantify it, but if you had to take a spectrum of polyps, that's the ones that turn into cancer most of the time. And so it's like you come in at 35, it's perfect. Take it out.
Yeah.
And you're done.
Yeah. And it feels so much better now. So basically, Daphne saved my life.
How was the recovery from that? I'm sorry to pass up the cute
colonoscopy?
Daphne saved my life. That was like, yeah, whatever.
Brush that over.
How long did it take to recover?
Oh, like nothing. I don't remember any sort of recovery.
It's a quick healing spot.
Yeah, most people are back to normal the next day.
Yeah.
Yeah, I was back in business.
How long does a colonoscopy take?
A lot of times it's between 7 to 15 minutes, depending on. It depends on how long it takes to get to the deepest portion of the colon, and then we usually come out for about at least 7-8 minutes on the withdrawal time. And then if you're taking samples, it takes longer. But for the person themselves, probably to get there, get the procedure done, talk with us and go, it's maybe like 45 minutes, an hour.
Wow, not bad.
So I think that we'd be very remiss without talking about diet. You've talked about the high fiber diet or regular fiber, which sometimes we think of as a high fiber diet. What are some of the things aside from paleo? So paleo removes dairy and gluten, and really beans as part of the paleo diet, that which is also a high fiber source. So I have a little bit of an issue with that.
Oh, they say you can't have beans?
You can't have beans. No.
You can't have beans.
Not on the paleo.
Doctor says you can have beans.
Yeah, so beans are one of our highest fiber, like by far the highest fiber foods that we can eat. So whenever you're teaching people about fiber, what are the top things, like what's the highlights that you give them to increase their fiber intake?
And so for the high fiber, definitely tell them to have at least one cruciferous vegetable a day. So, broccoli's easy, cauliflower's easy. Brussels sprouts, most people don't love, but I kind of tell them to start with those 3 because every grocery store has them. They're easy to prepare. I tell patients to try to add one fermented type food daily, whether it be sauerkraut or kombucha, which make sure it's low sugar because some of those are pretty high sugar.
I'm a kimchi fan because I'm a spicy
kimchi
girl.
or even pickles that need to be refrigerated where they don't sterilize it. Or a good source of fermented food. So I kind of tell them to start with that, and then to kind of build in and fill in with other stuff they like green leafy vegetables. Tell them to add lots of color like bell peppers, carrots, and stuff like that. And then if they do great with that fiber, Daphne, like you were mentioning, I like to add in the beans once they've gotten better, because a lot of patients come in eating 5 to 10 grams of fiber a day, and their main complaint is bloating.
Mm-hmm.
And so it's tough because I'm trying to ratchet up something that's going to potentially cause bloating, but in the long term, it's going to be great for them. And then the more they eat it, their body almost kind of starts to just crave it and expect it.
Mm-hmm.
And you notice when you don't have all those fresh fruits and vegetables. And so I start with those. I kind of tell them to set a goal of 15 grams for the first few weeks. And then I'll see them again in a few weeks and then ratchet it up to maybe 20, 25 grams. I don't love to add fiber supplements because I want them to get the nutrients from the fruits and vegetables. Right. I don't just want them to eat, McDonald's and then they take Metamucil or Benefiber. It's like those are fine in a pinch or in certain contexts, but I'm like, if you need the phytonutrients and the foods are already... The farming's done so poorly sometimes where the nutrients are so low, you have to eat like a pound of vegetables to get what you could 50 years ago. But I at least want them to get some of it. I don't want them to just get empty fiber. And so once they do well with that, when they get to about the 25, 30 gram point, that's when I tell them to try to substitute in some beans for some of their proteins and some of the things like that, especially if they can tolerate it, which most can since we've ratcheted it up like that. And then what kind of things do you do as well?
So as far as fiber goes, the average American gets somewhere between 12 and 13 grams of fiber a day. And the recommendations are 25 to 35 with, the higher you get, the better. And I've seen some studies that show that some of the hunter-gatherer populations get closer to like 40 or 50 on a regular basis. So, our body is capable, the human body is capable of having a whole lot more fiber. So usually I'll have them, I think of it like this. So beans are the highest source of fiber per serving, per half cup serving. And all beans across the board are similar. Then we have things like nuts and seeds.
But what's the downside to beans? Was it the carbs?
They have a song, beans, a musical fruit.
We've done this.
Yeah. Okay. So the downside is that it can cause gas and bloating.
Okay, but
As
so
far
there's
as
not
the
like,
paleo.
but there's not like there's too much sugar. Like there's not like an actual
They're
nutrient.
like, I think of them as thirds. So it's a third fiber, a third carbohydrates that can be turned into glucose, and then a third protein.
Okay,
So they have, it's kind of a combination, but it is considered a carbohydrate based food.
right. But you would claim that they're healthy enough to not
Despite-
worry
Oh,
about it.
absolutely.
Okay,
That's not where people are over.
that's not your problem.
Carbs.
Yeah.
No, yeah.
Um,
I like to see them try.
yeah,
Yeah.
right. So then nuts and seeds would be the next kind of category per serving, have somewhere between 3 and 4 grams of fiber. And then vegetables and fruits are on the lower side. So somewhere between 1 to 3 or 4 grams of fiber. So depending on what their average intake is. I mean, there are populations of people that are fast fooding it all the time. That's not the norm, I don't think. Normally, it's somebody that's doing like fast food occasionally. So they might be cooking at home or they might be going to a restaurant or something, but their actual meals don't contain much vegetable or fruit at all. I'll approach it a couple of different ways depending on what their diet is like. Either like, let's just look at eating the colors of the rainbow. So, and I'll go through all the different colors and what foods do you like that are red and what foods do you like that are orange and yellow. Have to do the Roy G. Biv every time. What kind of green foods do you like? And so kind of walk them through, like identify the foods that you know what to do with them. You know how to cook them. You're comfortable eating them. You could maybe enjoy them. And let's start with that first. And then we can add in, we can continually add things in with the goal of getting to that, 30-ish grams of fiber is usually what we shoot for. And if there are days where things don't go so well, and maybe you're like, it's been a really busy day, then that's where you can add in something like a psyllium husk or benefiber, or, we can add those things in as needed. But we really do want to start with the diet because, like you said, you're going to eat anyway. So if we up level the diet just a smidgen, continually up leveling it, not only are you feeding your gut microbiome, but your insulin resistance, your brain function. I mean, it's going to do so many other things than just taking a fiber supplement.
So obviously we could talk about this forever, and there's probably 3 more hours worth of topics that we could cover. So we'll have to have you back on. But before we let you go, what does living vividly mean to you?
I think, for me, the biggest thing that I've learned in this health journey is, trying to make all of this a lifestyle change, and not just working on one part, being like, I need to sleep more. I need to do that. I think it's good to attack things in little chunks and do that. But I think moving towards the goal of having like good emotional health, good physical health, good spiritual health, just it has to be a whole package. Because if one part of the piece falls, the whole system crumbles. And so I'm a big proponent of it's the whole picture, the whole person. And that changes through time. So I think when you think of that term, you want to think of how are you living your best version of yourself, the healthiest version, the happiest version, the best balanced version, the best father, husband, and doctor and person. So to me, I think of it as, working on all the little parts of your life to make it a package that's just beautiful and that just works well.
Thanks for listening to Living Vividly. If you enjoyed today's episode, please leave us a review and subscribe. We'd love to hear from you. For more resources and details from today's discussion, check out the show notes. Until next time, keep living vividly.